Okay. Yes. Hi, everybody. Hopefully you enjoyed this video of different chimeric animals. And today we'll be talking about some chimeric animals as well at our lecture. The last time we spoke about the code of life, and hopefully you remember something about gene therapies, about CAR-T. Hopefully you feel now much better and more comfortable discussing these topics. These are really tough topics. As I told you, if you manage to cope with life sciences, you would cope with any other industry. And today we'll be talking about even more complicated topics. It will be easier later on, but today we are diving into the most difficult part of the biotechnology space, and the most promising one. And so we'll be talking about living therapies, about actual living organisms or some living creatures which could be used as therapies, which could be created from scratch and used as therapies. And also we'll be talking a bit about multiomics as a major trend in life sciences. Again, I would like to remind you that global trends are very important, and if you want your company to have substantial value and to be sold to a big pharma player, you have to work and really implement some latest trends in your projects. And here you see that the major percentage of companies which were sold back in 2021 were early stage, really early stage projects with disruptive innovation inside. And of course, all the others, it's kind of a different story. So primarily, big pharma players and generally corporations, they acquire great science and great innovation, and they can do that at very early stage when there's even no clinical data, or just a little clinical data. Still, they can buy the company. So this is the overview of the most trendiest, the hottest areas of biotechnology space. Last time we talked about CAR-T, we talked about whatever it is. I also would like to just to briefly touch base on Rna Therapeutics, which is a super hot area as well. But you see fewer companies here. But anyway, the RNA therapy is probably one of the hottest areas right now. And then we'll talk a bit about stem cells, which are not mentioned in here. And that's why we'll be talking just really shortly on that. And finally, the topic of today's lecture would be synthetic biology and microbiome. We'll also touch base on multi-omics and DNA storage. Okay. Yeah, so this is central dogma. Hopefully everybody is now familiar with that. They know what is central dogma. When was central dogma kind of formulated? Which year? Which year? Last time I mentioned the year of 1958. Okay, that's good. Okay, when was the DNA discovered? I mean, the structure of the DNA. Which year was this double helix? Which year and who did that? What's the name? Rosalind? No? Rosalind was one of those people who unfortunately didn't get the Nobel Prize. So, okay. Now this is something which everybody should know, especially if you're somehow dealing with life sciences, but it's a general knowledge. So yes, so James Watson and Francis Crick, yes, these two guys, and Rosalind, who were never mentioned, unfortunately, and well, she was actually, but there was a bit complicated relationships between all of them. But the two guys who got the Nobel Prize were Jim Watson and Francis Crick. Okay, so this is the DNA. This is the central dogma of molecular biology. It says that there's DNA DNA can replicate, so it's replication. DNA can be transcribed into RNA, so it's transcription. And then RNA can actually be translated into a protein. Protein, this is the polymer of amino acids, which forms some kind of shape. So this is, I would say, like hard drive. This is like RAM memory, and this is already a program which is being realized. So this, you have to remember that. We'll be discussing this at length at our molecular biology course if you take it. But anyway, now a few words about RNA therapies. Just a few words. RNA became really like famous through the obviously RNA vaccines. But the whole idea of RNA is kind of that if you change the DNA, it's a change which you normally cannot undo. And that is why currently we don't have actually tools to change back the DNA if we edit it. We don't have even really accurate tools to edit DNA. So that's why it is really very risky to change our genome. RNA doesn't have this problem. RNA technologies based on this, like on that they can actually stop some RNA or inhibit, I would say, or repress some RNA in terms of their translation into proteins. So we can apply a drug which would stop translation of specific RNA into a protein, which essentially means that you can switch on and off a gene without altering the genome. So you can actually really intervene here at RNA stage and just stop this RNA from translating into actual protein. And this could be a molecule which will be very specific, which will target very specific RNA and stop it from being translated. This is one way how you can make RNA therapies. The other way, in RNA vaccines, that's totally different way. You're creating an RNA which you deliver into human cells, and then within human cells, these new RNAs from the drug get translated into new protein. And if this protein is an antigen, as we have an RNA like vaccine in Moderna, so they code the code of virus, the code of the spike protein, spike protein on the coronavirus. That's why it's called corona. So the spike protein, they code it with an RNA, delivered it into our cells, and the whole human becomes a plant for producing these antigens which immune system recognizes and creates immune answer and immune response to it and remembers that. So this is RNA, and this, as we see that initially there were specific small RNAs which are actually used to repress and stop RNA from translation. These were small RNAs, but at that point the more trendy are messenger RNAs, which actually code proteins. And the RNA vaccines are a great example of that. So this is now more trendier to create products which are based on messenger RNA, on delivery of the whole code of a specific protein to the cells and producing this within a human being. Okay, so this is the RNA. Hopefully understand the mechanism of action, how it works now, and we are done with RNA. The other thing, the other technology, which is a stem cell technology, and unfortunately, at least I'm regretful on that one, that these are not that trendy so far. And this is an approach where you take stem cells which can differentiate into any cells. So the idea is that you have a specific tissue or a specific organ which is damaged, and then you can take the cells, the stem cells, put this on the organ, and the stem cells differentiate into the organ tissue. And this would actually happen, and in some experiments it happens. but there are so high regulatory hurdles for that it's one thing, and technologically it's not straightforward, and from the scaling perspective, and scaling is very important, as you remember. So from many perspectives, this is still an early stage for this type of technologies. We're seeing many more companies entering the space, but we haven't seen really a great success, like a multi-billion company in the space anyway. And there are companies in the space which are actually late stage, doing their trials in like phase 3, like some final trials in humans, and still are very low in their evaluations. Anyway, so this is stem cells. The story here is that initially the stem cells, the actual patient's cells were used as stem cells. And this was very like difficult, or they were using embryonic cells also of patients. So this is like a very difficult source for cells. And finally, in 2006, there was a discovery that you can take any mature cell from the body, and apply specific factors on that, and then like move the cell into the stem cell stage. So you take the mature cell from like part of your body, and then you can make it into like a phenotype, the status of cell, which is a stem cell. So, and this was amazing. So the scientists who discovered that got their Nobel Prizes for that, the Nobel Prize for that. However, it is still not being used. And by the way, if you heard about that, there is a 3 billion startup just now launched, Altos Labs, which is using this kind of technology of trying to get back our kind of mature cells into their early stage and potentially radically expand the human lifespan. Have you heard of Altos Labs? I think just yesterday in Forbes there was an article by my friends who wrote about that. Okay, I will send it over the chat. So it's pretty interesting. Anyway, hopefully this will be the first major startup in the space. At least with such kind of funding behind, they may succeed. So the primary topics of our today's lecture would be the synthetic biology, microbiome, and multiomics. And here there are already some successes. Synthetic biology, Twist Biosciences went public, and there are some controversies around that, and they probably oversold what they have potentially. But you should read the papers, and again, hopefully, there's such an effect when you hear something in the lectures. Then the next day or the same day you will see this in the newspapers and in the internet, probably because your phones are listening to that, and adding like on all of your social networks some news about that. So hopefully we'll see about Twist Biosciences, because this is really like a controversial situation. But the Twist Biosciences by itself is really like brought a lot of promise, and they were like early pioneer of DNA synthesis. And DNA synthesis, like creating DNA from scratch, is one of the, I would say, enabling technologies for synthetic biology. Ginkgo Bioworks is another story, funded by Bill Gates, not mentioned here, but anyway, they are trying to redesign living creatures as therapies. So we'll go very deep into synthetic biology just now. Microbiome is a story of using our bacteria, which are living in our guts and generally in us, and using them as therapy. So they are actually living creatures which we can use as therapy, and we'll be discussing some of the companies like Ceres and the other one which talks about interspecies communications. And finally, multiomics. Multiomics is a different topic, but it probably summarizes some of the previous. So essentially, it is like summary of all the kind of like all the genome, transcriptome, transcriptomics, all your RNAs which you have in you, and then all the metabolomics, all the whatever, um, all the microbiome, etc. So all of that are omics. So you have this genome, genomics, proteomics, all the proteins. So all of that, if you combine that, then you can actually get some new information which you'll not be able to get from one omics, from one like genome. That's not enough for you to get, to give you a diagnose, or to like to find out that early cancer signs or anything like that. So this is multi-omics, which is primarily early diagnostics. So why we're talking about synthetic genomics, synthetic biology later than gene therapies and many other things? So like conceptually, the DNA test, like DNA test means that you are capable of reading DNA, right? But then DNA editing, like gene therapies, which we discussed before that, this is about editing DNA. But then the question is whether we can actually write DNA, whether we can not edit like some living organisms, or whether we can create organisms from scratch. And this is a question where we have already the answer. And yes, the answer is yes. We can actually engineer life. We can engineer real living cells. We can engineer bacteria. We can engineer potentially some like multicellular organisms and create them and make them compatible with human organisms if we want to transplant organs from an animal to a human. And so this is all already possible. It's happening right now. It's the hottest area, one of the hottest fields in the life sciences space. But why it happened later than DNA tests? Of course, there's a very simple explanation to it, because the cost of DNA reading went down dramatically over the last few years because of new technologies for DNA sequencing. It's called new generation sequencing, NGS technologies. So because of NGS, this logarithmic scale here, so it fell dramatically. And now, hopefully, we'll experience the same with the DNA synthesis, with writing the code of life. Currently, you see here that even if there would not be a dramatic change, we still will see substantial decline in costs for synthesis of DNA. But if there's a second generation of DNA synthesis, as we had with the next generation sequencing, then we may be surrounded by creatures which were synthesized and created from scratch. And we'll be discussing this today at this lecture. Hopefully you've seen the video which I showed to you before this lecture, showing many different chimeric animals, combining actually fish and cats and dogs and octopuses. So yeah, so let's look into synthetic biology. Synthetic biology is a multidisciplinary field which includes everything: biology, engineering, informatics, data science, and in order to do anything in synthetic biology, it requires all of these technologies. So there's a kind of technological stack specific for synthetic biology. And there are many things you can do there. You can do you can synthesize DNA, for example. You can automate synthesis of cells. You can produce some proteomics, something which would regulate these cells around that. So you can create reagents. You can do anything. So the synthetic biology space is very large from the technological perspective as a technology stack. It has a lot of ways how we can contribute to that. And yeah, so here you see essentially that you kind of creating these DNAs. You can put this into cells, and this will produce something, etc. So yeah, this is a good summary here. So essentially, from the technology perspective, for you to be successful in synthetic biology, and that's why it's really a tough field to be specialized in, you have to combine all of these particular expertises, like including chemistry, bioengineering, bioinformatics, biology, etc. And then with that, you can create some living systems, you can create sensors, which would be very specific. You can make other components which can be used in many, many industries. And this is important that the kind of gene therapies and many other things, they were sometimes used in agriculture, but synthetic biology is straightforwardly used in many other industries, not just in medicine. By far not, not only in the medicine. So you can create therapeutics, biopharmaceuticals. You can most of the investments, by the way, are into this space, into medicine and food. With food, yes, you can create food from scratch without killing an animal. A lot of synthetic biology is used in the agricultural sector. You can create bioenergy and many other things. So this is really a disruptive technology for many industries. And through synthetic biology, biology in general and life sciences can get into many industries and disrupt them. There are specific products which you can get for different industries. Like for the medicine, there's CAR-T. You remember CAR-T? So this is also synthetic biology is being used to create these specific T cells or like different types of cells, and and many other things. So you can create programmable organoids, and today we'll be discussing how it actually works. For food industry, you can create artificial meat. You can make food substitutes or pet food, food biosensors, right? So whatever you have to work with actual life, measure it, or react to something in the living organism. You can use synthetic biology and create so-called, I would say, nanorobots or some systems which will react in living creature and produce something. So like on energy, you can create specific bacteria which would which would digest something and create biofuel through that, or could be enzymes for some specific chemical reaction or anything like that. So there are many applications here, and this is, I would say, a very far-reaching technology as compared to some others which we'll be discussing during this module. The synthetic biology is still very much underappreciated and undervalued, and there are many companies in synthetic biology which are already listed and public, but the valuations are not that high. In terms of technologies and specific applications, this is a list of specific, I would say, more narrow ways how you can apply general concept of synthetic biology and which things you can do from the scientific point of view. This is based on the publications, and publications are normally a sign that something is being explored, and something at some point may become a technology and translate from a fundamental science into something which you can use in your product. So these are for example, cell engineering, like creating actual cells with some programs inside. This is already potentially possible, but you see, it says that there are not too many publications so far on that topic, but the speed of the growth of the volume of such publications is very high. It means that something is like brewing here, and you have to monitor that space, and hopefully something will come out of that, and you could use this to meet some really unmet need. to make it more clear to you, synthetic biology, like conceptually, in a more straightforward fashion, is the following. You have code, like in the previous lecture I told you about the code of life, right? It's DNA or RNA, whatever. So you have a code which you can, there are like different ways how you can write it. And this code you can put into some living either cell, or you can put this into a virus and, and deliver this into a human or into an organism so this is kind of hardware, this is software. So you write code with some circuits, some, if, then, else, and I will show you how it works in biology. So you write if, then, else, if something is going wrong, then, produce something so that it gets better within the human. So if something is kind of like not some protein is missing in the human body, so you put something and it checks if this is, so you actually put this code into a hardware, which could be a cell of like T cell, if it's like immunity, or it could be a stem cell iPSC, which I mentioned today, which is a stem cell which is produced out of a human, out of a mature human cell, or human stem cell, which is an embryonic human cell. Or you can put this into a virus and deliver it into a human in that way. And then this code would be doing something within a human being, within an organism, and can actually potentially monitor or cure some diseases in oncology space, immunology, whatever, regenerative medicine, neurodegeneration diseases, and many others. So this is the way synthetic biology works. Hopefully I think this is the most clear slide. Did you get that? Hopefully you got it. Okay, so hopefully you're not lost so far. And now, just showing you how you can actually code with DNA. So this is an example. If you want to code, you have to replicate somehow in the biological systems the kind of the circuits, the gates, the logical gates which you have in computers. In computers you have on, and, or, you have XOR, okay? You have NOT, right? So NOT gate means that if you get 1 as your input, you get 0 as your output, right? So it means NOT gate. So you have to somehow do the same thing in biology, and it's kind of not straightforward how you do that. But this is something that shows you how you do that. Hopefully you remember, I mentioned that like last time that within 5 minutes you will become molecular biologists, right? So you remember that this is like DNA, and the DNA is number of genes coded there, and each gene has a special place where it shows that here you have to start reading me. So the specific code of DNA which says here comes the gene. So next to this code comes the gene which you have to read and to kind of transcribe first into RNA, and then through RNA to a protein, translate into protein. So this is called theta box. There are promoters also, promoters which say that, please, let's produce this gene more of this gene or less of this gene, depends on the promoter. So you can actually code with DNA the following. So for example, it is actually one DNA, it could be actually very long, but at some point there's a DNA which has a gene 1, and once it gets transcribed into RNA and then translated into the protein, then this is protein 1. And then there's another place which codes gene 2, and this is protein 2. And then when they combine, then they bind to this promoter, and then the polymerase, this is like special kind of vehicle to start reading the gene. It knows that it has to bind here and start transcribing this gene. So unless 2 of those proteins are present, it will not start start producing these RNAs and then the proteins. So it means that's and. So they both have to be here so that it starts producing. And here you also have some signals. So if something happens and you get two signals, so there are, say, two proteins produced in the cell, only if the both proteins are present in the cell, then you produce this new protein. Do you get that? So this is a logical circuit of a computer which you can replicate and do the same thing in a actually living cell and code this into a DNA. The same thing is for OR. So there could be some two promoters, and each of those promoters, each of those activating promoters, can make the polymerase read this gene. So either this is enough to start producing this gene or that. So this is OR. And NOT, it says that if both proteins are present, then please stop producing this gene. So what it means is that you can create a bacteria which will monitor presence of two proteins. If these two proteins are enough, or they are substantial enough in terms of their concentration, then they may stop bacteria's production of specific gene, of specific protein. And if some of those proteins are missing, then this bacteria will start producing a protein which is somehow missing in the organism, and which may actually, this protein may alter something and may I would say drive the production of this particular protein, so that it stops. So it has like feedback loop here. So this is really amazing. This is possible in the biology space, and this is called synthetic biology. So you can create life, you can program life, would be a living therapy, which would monitor some specific proteins within your body and will only produce the necessary molecule, the necessary potential therapy, only if something is missing. And synthetic biologists managed to create 16 up to like 20 steps within bacteria, so that they're kind of a real program within a bacteria. You can produce that, you can create a DNA, and you can actually do many other things. So hopefully now you understand the principle of synthetic biology. This is really amazing. So you take, this is a software, you put this into either a cell or you put this into a virus, and this code gets into the right place in the organisms, into the organism, and produces something where it's necessary. By the way, I think here, I would definitely add bacteria. Bacteria is missing here. This is from one of the companies. This is a slide from one of the companies which is really doing great things in synthetic biology. Anyway, these circuits are already known. They are all put online. There's a website with the registry of standard biological parts, and it's called BioBricks. So you can really program your ideas, put this you can do this as modules when you have in the programming. You don't program it from scratch. You normally add some modules from here and there. Here you have all these modules, and you can create your program easily, even if you're not the best programmer and you don't have a way to test each of those circuits, but you can find already somebody else may have done that, and you can do that. So the problem with synthetic biology is that it's unclear how to make it as predictable as possible. And one of the ideas was to make an empty cell without genes, and find a way, find the kind of the minimal cell. So the minimal code in the cell, which is enough for cell to replicate, to be alive generally. So this was the challenge. And there was Craig Venter. Anybody heard of Craig Venter here? Craig Venter. Craig Venter. So Craig Venter is like Elon Musk in the kind of in every other industry. So he is like Craig Venter in biology is like Elon Musk in whatever in the space in other industries. So Craig Venter became famous because he was like competing with a state project called Human Genome Project. Human Genome Project you probably heard. I told you like in the last lecture about Human Genome Project. So where they tried and like to to read the whole genome of human being. It took many years, and the work was finalized in 2003, exactly 50 years after the discovery of the structure of DNA. So DNA of the human being was kind of fully read. And in the meantime, Craig Venter was doing the same. He tried to read the genome, but he did it 10 times less expensive than this was done by the state. And he showed that he can do this quicker and cheaper, and that's why he became quite famous. But later on, what he did, he started to do the minimal cell. So what he did, he took the cell with all of its DNA, and then started to just take out a gene and check if the cell is still alive, and going like one by one. And finally, at some point, he arrived at a cell which was like around, I would say, less than 400. I think less than 500 genes. So less than 500 genes seemed to be enough for cell to be alive, to replicate. He did this totally neutral, like brute force. He didn't understand what the function of each gene is, but he was just deleting one by one and checking if it works or not. For this operation, of course, he need a new mechanism how you synthesize DNA, because like in order to actually check each combination of genes, he had to create a lot of cells, a lot of cells. So for that, he was one of the pioneers of synthetic biology. He created a company, Synthetic Genomics, which is now a famous company. He also created human longevity story. It's another story, but Synthetic Genomics is a company where they actually create chimeric animals which will hold human organs for transplantation. And they're doing many other things. But synthetic genomics is based on the inventions which were done by Craig Venter when he was trying to identify this minimal cell, and now they can produce DNA from scratch very quickly and efficiently, and a very long DNA. What is interesting, when Craig Venter did that, he was not sure whether it would actually work, because you take the cell, which is kind of live, but then you take out the DNA out of it, and normally it's kind of like it doesn't work anymore. But then if you put the synthesized DNA, which is definitely not a live molecule or live organism, what would happen? Whether this cell with a completely different DNA would it actually work, and would it actually be alive? And he tried this out, and yes, it is a live cell. With the DNA synthesized outside of the cell, and replaced when he actually replaced the DNA which was initially in the cell. So it is possible to take a living cell, empty it, so take out the DNA out of it, and then create DNA and put this into this artificial cell, and this cell would be alive. Now the challenge is whether you can do this like not top-down approach, but bottom-up, whether you can create DNA, create membrane, create nucleus, create all of these non-alive things combined, and that creates a living cell. So this is the challenge, which is still doesn't have an answer, but it feels like it's all pure mechanics. So it's just mechanics. It's just chemistry, physics, nothing special. So most probably, at some point, we'll be able to assemble cells ourselves. So we'll synthesize DNA, we'll synthesize all the organoids of the cell, and then assemble it from scratch. We shall see this. This is the challenge of the synthetic biology, the next challenge. Okay. So in terms of investment, I mentioned that the synthetic biology has immensely broad applications, and we see this in this chart on the right. By the way, this is a very recent chart. This is I will send to you the report by synthetic it was previously called SynBioBeta, but now it's called it's built with biology. It's a website where they track all the synthetic biology stories. So they just 2 days ago they published their latest report on the state of the industry for the whole year of 2021. And what it shows here is that health and medicine is still the largest chunk of investments in synthetic biology space, but food and nutrition generally is kind of the second, and then organic engineering and some others. But you see the spectrum is very broad here, and these are investments. So investments, these are not sales, not like capitalizations, but this means that from these investments we'll see a lot of companies coming later on in a few years. So it's kind of early traction. And you can see what will happen in these industries in 5 to 7 years by looking at which specific companies investors are looking at and investing. Yeah, and then of course there's a stack here, technological stack. So there are many things you can do here, and many companies who are generally becoming large, like creating the infrastructure for synthetic biology and infrastructure for startups and for the industry. Okay, so this is the investments. They are on the rise because of this broad spectrum applicability. You have many new investors who would normally not invest into life sciences space. So you would have the IT companies are now more and more investing into synthetic biology because they kind of understand that this is so much engineering and so much of a concept very close to software and hardware that it is no longer life sciences, which was unpredictable, very stochastic, which was kind of 50% patients react and the other don't respond. So here with bioengineering, you know specifically who will react, respond, and why, and what is happening in the organism at the molecular level, and you can produce by molecules by rationally designing them. So rational design is one of the major changes. So you really produce the exact molecule or the exact cell which will have a specific function and will cure fundamentally the disease because it actually is like kind of a live-in therapy, fully autonomous, personalized, and all the kind of trendy words. Okay, so these are the IT companies. They are investing heavily into the space. And the other are venture funds, of course, but they are doing primarily like investing in the medicine space. There are some corporations who are like investing, and they are specifically looking for technologies which may disrupt their work. So like it could be oil and gas companies who are looking to produce biofuels, or not biofuels, but potentially in oil and gas they can find a way how they can produce energy from some waste of their production. The same thing goes for agricultural industry. You can actually code a bacteria which will do something with your waste and something which is useful for you. This could be energy. This could be anything else. So in like without that you would be kind of throwing this junk away, this waste. But here you can produce something out of it based on specific bacteria. And then, of course, there are a lot of state programs around that. And yeah, so all the countries are kind of launching their own programs to support synthesis or synthetic biology generally, because this is a really disruptive field. But the problem is that not not everybody knows the synthetic biology. Have you heard of synthetic biology before this lecture? Who have you heard of synthetic biology? 1, 2, 3, 4, 5. Okay. Okay, good. Okay, so it's like we are better than there's only 75% who never heard of that. So probably more than 50% here heard of the synthetic biology before my lecture. That's great. So 75% of people never heard of synthetic biology, and they are quite concerned because this is something they don't know. And then, like 50% think that this has to be supported by the government, but this is from those who know what it is. But then, of course, there are many people who are very concerned and asking that this gets first very tightly regulated and then being developed. Yeah. So anyway, you have to to look into that, and now since you know now what is synthetic biology, that it's really a great technology, you have to support that. I would say that there are huge risks about synthetic biology anyway, but yeah, so it's still quite a regulated field, and hopefully we'll not see of that, risks being realized. Yeah, just another example for you, and we'll be discussing this at our seminars, but anyway, and another example of synthetic biology is a company called Proteome. So what they're doing, they are changing the mosquitoes who carry the malaria disease, and through changing them, these mosquitoes, they would not any longer carry malaria disease, and through that, if these mosquitoes replace the natural population, the wild-type mosquitoes, then we can cure malaria, right? So we can get rid of that. And so this is just an example of a kind of a therapy, which is a totally different way how you can cure a disease. And you know that malaria is one of the deadliest diseases and one of the worst diseases, not on the developed world, but in some countries this is really tough. And if this is the way you can actually cure this disease, this is amazing. But the question is, when you already work in nature, and you're changing something within that, you don't know what could be the consequences, because this is a system, and somebody feeds on this and whatever, and there's malaria, which is kind of required for some ecosystem balance. You never know, but hopefully nothing will happen, and we'll get rid of malaria. So, but what is important here is that this is not science fiction. This is already happening. This already got approved in Latin America, and this is being used already, in reality. So this is not something which is kind of science fiction. Okay, so now just a summary of a few points from this Build with Biology report. You can read it by yourselves in the report, but I'll just go quickly through that. So what it says is that that in terms of food, essentially there are many things which you have to take into consideration if you would be doing a startup in the synthetic biology space and synthetic food specifically. Initially there were many concerns that this would be unnatural, artificial, but now actually we're trying and making not only plant-based meat, but also cell-based. like cell meats. So based on, take the cell from a cow and actually grow it, and this becomes a meat which is normally even kind of healthier potentially. So hopefully we shall see that, but definitely more ethical. And ethics here is the last point here, and of course, um, I think this is a major consideration for many people. They would actually enjoy more kind of this plant-grown food or lab-grown food, lab-grown meat, because no animal is slaughtered. And again, I mentioned that it's potentially healthier, no antibiotics, nothing like, it's just pure meat, hopefully pure proteins, and also potentially more tasty, hopefully. But we shall see that. But you probably, did anybody try alternative meat here? Just one? Anybody else? Oh, Beyond Meat. Okay. Which? So you tried this? Did you try this by yourself? Yeah, I just know the company. Okay. Okay. So, but yeah, Beyond Meat is obviously the most like famous company doing that. Um, well, we actually have our actually master student, and she is actually doing the plant-grown, not plant-grown meats. She's doing, um, uh, so she's just using some microorganisms to produce whatever, like cheese, milk, potentially meat. So we shall see how it works. Probably she will have a multi-billion startup. We don't know yet. Um, yeah, so this is potentially healthier, hopefully more delicious, and so there's no, I would say, no sacrifice. Normally, healthier food means sacrifices, right? So hopefully with that, we're kind of having healthier food without sacrifice and more ethical food. And of course, there's a lot of pressure now from the ESG agenda that producing meat is very bad for ecology. It's one of the major sources um, for the hmm? For gas emission, right? For the CO2 gas emissions, right? So, and this is like one of the major reason for that. So one of the major thing. Hopefully, with that, we can do this not only environmentally sustainable, but also beneficial, right? So this is a different story. So it's not about even sustainability; it's even good. So this is to finalize about synthetic biology. Now you hopefully have a grasp of the field. There's a great course on from MIT on edx.org. I will send you the link. That's a great course on synthetic biology, showing you all the circuits, the ideas behind that, and it's a very tough course. Now microbiome. Microbiome is sometimes very much, I would say, unappreciated. And we will be talking like the microbiome is everywhere. So microbiome could be, say, in the ground. So you have in the ground where kind of, and you can by microbiome by those bacteria which colonize this or that ground. You can see which particular bacteria are there and understand whether the ground is healthy or not, or unhealthy, kind of, and whether it will produce plants or not. Same thing in the human being. So you have a microbiome in oral space, in the in urogenital microbiome, and then the gut. But the major focus is gut microbiome. This gut microbiome, it feels at least that it may drive many diseases. Even something where you would never think that this may be somehow connected with your bacteria. For example, some psychology problems, or like depressions, or schizophrenia, or something related to oncology. It's really difficult to connect oncology and microbiome in the gut. You think that the gut would be mostly about some metabolic diseases or like GI tract diseases, something like that. But actually, you know that in oncology, the very much very important role plays the immune system. An immune system is potentially largely driven by your gut microbiome. So it's all kind of interrelated. So, and here, just some basic facts. So there are, I would say, at least the same number of cells for microbes as to cells of the human being. And there are, by the way, different numbers. I've seen 3 times more than the cells of human being as well. So we have like around 30 trillion cells in our body, and then the number of microbes is 100 trillion, whatever numbers. So anyway, it's 1 to 1 or 3 to 1. So at least they are probably more us than we are. So kind of at least from the number perspective. Also, it's 3% of human body mass. Not so much. They are generally bacteria are much smaller, of course, than human cell. Anyway, interesting thing is that if you look at all the genes which are in all these colonies of this species, of this bacteria, it turns out that there are 10 times more genes in these bacteria than the genes in the human being. And how many genes in the human being? Last time I mentioned that. No? So how many genes do we have in our DNA? Okay. Okay, I'm just checking how you kind of remember anything from my previous lecture. Probably almost nothing anyway. Probably too much information. Who knows? So 20,000 genes. There are around 20,000 genes in the human genome. Initially there was we expected there would be 100,000, but then it turned out that it's many less. And there are even viruses or bacteria or kind of other species which have many more, or plants who have many more genes than human beings. Anyway, and these genes in these bacteria, obviously they also get produced. They produce RNA, which is being translated into proteins. Or are RNA by themselves are also doing something. So you have a lot of RNAs, a lot of proteins, which are being produced by microbiome, by your bacteria, right? And you know that both the RNA and proteins are functional and can do something to your health inside your body. So, which means that you have to take into consideration what these bacteria are producing, whether they are not producing anything harmful, and of course, I mentioned to you that there are a lot of association with different disease states. Of course, infections, GI tract diseases, and then metabolic diseases like diabetes, allergies, so any immune diseases like allergies, asthma, and others. All of that potentially could be driven to some degree, or at least diagnosed through our microbiome. So the way it works, it's still science, still a bit of science. So microbiome is a system of different colonies of bacteria. And you see here, for example, the urogenital kind of microbiome would be different than to your oral space or your whatever gut microbiome. It's kind of very different. And what is interesting here is that normally the microbiome is kind of stable and keeps the proportion between different bacteria the same by itself. But through some intervention, you can move this state to the other state. And potentially, this state could be a disease state. And if you add, this whatever green bacteria and yellow ones to this microbiome, then it moves, through that and moves to another balance, another new balance. And if this is kind of a healthy state, then the person potentially is cured. So we don't know yet whether microbiome is a reflection of your disease state, or it actually drives your disease state, or it kind of complicates the disease. But we're just figuring out right now, and there are many startups around the space who are trying to figure out what actually depends on what. The typical way, the most kind of typical way, how you work with microbiome, you actually take specific, I would say, bacteria, and there are a few ways how you can do that. So you take a set of bacteria, you put this into a human, and potentially, hopefully, change the composition of microbiome, and then there's like change in function of the microbiome, and hopefully through this you get change of function of the organ. So this is kind of the way microbiome works, like in principal terms. And these were the first therapies. That's what they were trying. The company called Rna Therapeutics with the ticker MCRB, microbiome, MCRB. That's what they were trying. This is the slide from their presentation anyway. The the story was that some people they took too many antibiotics and their microbiome was damaged, and at some point the microbiome could not get into the right state, into a healthy state, and this specific bacteria called Clostridia difficile, or C. diff, like the short term C. diff. So this bacteria would colonize all the space what is available. So if you drink too many antibiotics and destroy your microbiome, the C. diff takes the place of all the bacteria which are wiped out. And then when you after you get the C. diff and your microbiome is wrong, then you kind of start to have really big problems with your gut. So you have diarrhea, you cannot eat, you cannot digest efficiently, and it's clear that something is wrong with your microbiome, and you get normally before that you would get from a doctor another set of antibiotics, and you would drink these antibiotics which will kill C. diff and kill others, but C. diff, this bacteria, they would get for some time into spores, and once you stop drinking antibiotics, they will get back again and may multiply even more. And you get again a new set of antibiotics, and then you're getting the C. diff into spores, but you're killing part of your microbiome as well. And C. diff again colonizes all the space. And at some point, it's just too late. There's no other drug, and C. diff becomes resistant to all of these antibiotics. And people are like really dying out of that because they cannot digest food. They are like dying. Over 10,000 people in the U.S. alone are dying from that because of the overusage of antibiotics. And the only way how you can cure that, and that's not like if you wanted to have a dinner today, probably not the best topic, but the only way to cure that was to transplant, to make a fecal transfer from a healthy person, take the stool and put this into a patient, and then the microbiome of healthy patient would actually recreate the whole healthy microbiome, and again your gut would act appropriately. Your organs will act accordingly and fine. What Rna Therapeutics tried to do, they instead of fecal transfer, they found a way how they can actually find a combination of bacteria, not the whole microbiome from healthy patient, but it's enough to take just few bacteria from, just few bacteria, produce them, not take from fecal transfer, but just produce them, and then put this into a capsule, drive them, move them into a spore state, and put this into human being, and then change the microbiome and get the person healthy without fecal transfer and generally without new sets of antibiotics. So this was an amazing technology. And they actually failed in phase 3. Now they have redone their phase 3, and hopefully they will be successful. So please check the MCRB ticker. Okay, there are other ways how you can approach that. Ceros, by the way, here. So other ways are that you can make some like beneficial bacteria, or you can find a way how to protect beneficial bacteria, or you can create this life biotherapeutics. So you can put actual bacteria uh, and place them into your gut, but it's difficult to put them at the right place and really to move it through like all of our systems and make sure they are not digested or broken down. And of course, somebody asked me last time about phage. There are phages who can potentially attack bad, microbes, bad bacteria. So, like, who asked me about phages? Well, okay, finally, yeah, I think this person here. Okay, so but the the problem with that approach is that potentially it's not about killing bacteria, but it's about making a balance. And potentially, it's better to kind of feed some bacteria so that they kind of grow, so their colony grows, and if they are beneficial, or if the balance is getting better, then you can get beneficial results for the health. And it's probably worse to actually kill bacteria because it's kind of more complicated. However, bacteriophages are very, very specific and they kill very specific bacteria. They do not infect, like broadly bacteria. They infect very specific bacteria and kill them. Okay. So, in terms of investment, the microbiome investments are generally growing. Again, this is a hot area, hot space. Here is the venture funding for the last years, and yeah. So this, I would say that back in 2015, this was probably a hotter area. Not in terms of the number of investments or the amounts of investments, but currently there are already like too many companies in the space. They're doing different approaches. Some of them is, by the way, not pure kind of medicine approach. So they are like doing a kind of a food approach or probiotics or nutraceuticals approach, because as I mentioned to you, it's potentially better to either add bacteria or feed bacteria in your gut to rebalance the kind of the proportions of the colonies. Generally speaking, I didn't mention that, but generally speaking, there are around 300 bacteria. So around 300 types of bacteria in our guts. So there are like not thousands or like millions, whatever. So it's 300 types. But then, so you can actually find the right proportions potentially. So basically you kind of, if you manage to engineer ideally, and you remember that ideally we make a bioengineering approach to everything we do in life sciences. So if you are capable of engineering a specific proportion between the different bacteria, between different colonies, and by that you can actually test if this cures the disease, then this would be an amazing startup. So, and there are startups doing that, trying to engineer a specific microbiome within a living organism, within a human being. So we shall see who of them would succeed. But it's, of course, it's much more complicated task than probiotics or anything like that, or nutraceuticals. Okay, so this is the story of microbiome in terms of investments. So there are now like actually, as you see, like 64 companies mentioned here in the database. So it's like 60 companies. It's not hundreds of thousands, and they have like 150 like potential diseases, potential indications. So it's a broad spectrum technology, and it's new modality. Again, so what we are discussing here, there was like 5 years ago, 10 years ago, there were no drugs like that generally, and definitely not drugs which were approved. No gene therapies. First gene therapy was approved 2017, as you hopefully remember from my previous lecture. And microbiome therapies are also being like approved in these years. And finally, I want to finalize the microbiome talk by mentioning another company and another like next generation modality in microbiome space. So something for the future. It's not something current, but it's even like further away. The idea is that we can combine the microbiome. This is the bacteria. Our system biology, so systems in our in the human body, and the plant system, so whatever we eat. And if we find a way how we can integrate all of those things, then this could actually work. And this is already called interspecies communications. So interspecies communications is kind of science fiction, it's a philosophical statement, but now it's actually translating into actual medicine. So, for example, you can use the vesicles which are in the plants to put something, potentially a bacteria or something, into the human body, into a cell of human body. Or you can, for example, there's a risk, for example, that you're taking some pills, and these pills by themselves are feeding some specific bacteria, and bacteria, their colony is rising, and that disturbs your microbiome, and this bacteria are actually producing some toxin, or at least toxin at these amounts, right? So at the scale, and then you have side effects of specific drug, and you don't know why, because you don't see a specific reason why would this particular molecule get some side effect on your heart. But it turns out that this kind of pill actually drives a rise of some colony in your gut of some bacteria which are producing toxin, which is somehow brought into which is somehow affecting your heart. So you have to think about all of these in the future. So you're not producing just one molecule or pill, but you understand how this will be digested, how this drives your microbiome, and then which means bacteria, which bacteria will actually rise in their colonies, what they are producing, how these proteins are affecting your organism, etc. So this would be something new and something more kind of transformative if we could combine all of these things in a potential. And this is actually a real company, also a slide from a company which we're evaluating and will potentially invest. So it's probably next generation of microbiome. But this is just one company. It's not yet a real major trend. But the idea of interspecies communications is really very impressive to me. Okay. And finally, the last, the one but last topic for today is multi-omics. So these genetic tests, these are really funny thing. So genetic tests essentially show you the potentially some predisposition for specific disease, for some state. And normally, so you for example, you have here the kind of the genome, and ideally, if you're talking about genomics, you would analyze not just a few genes, or you would analyze whole genome, and it's called whole genome sequencing. Most of the tests which are currently available for consumers today, they are sequencing not your whole genome. They're just doing just either specific genes which functions are known, or the best case, they are sequencing all of your exons. Exons are those genes which code proteins. So they just, they essentially sequencing and and covering all of your exons. Exons are those genes that code specific protein. But then there's a procedure called whole genome sequencing, WGS. And then if you do the whole genome sequencing, or you at least your full genome in terms of or it's called exome, if you sequence only all your exons, you can actually do some kind of relationships between these genes. You can try and find some ideas how they interact, but not much. And so the same thing is for proteome, for example. Proteome is all the proteins in your bloodstream, for example. You can analyze those as well, and then you can analyze all of your metabolome. It's all the metabolites, which are essentially products of any chemical reactions within your body. And then, of course, you could check all the RNAs. It's called transcriptomics, transcriptome. But of course, you would make a much better understanding of the state of the human organism if you could combine all of that and find some real interaction. So there's a predisposition in your genome for this particular disease, but from the RNA, it feels like this particular gene is not expressed, right? But even if this is expressed, but somehow at RNA level, there's a way how RNA is being spliced, how it's being interpreted, and how it may actually code different proteins, not just one, but different proteins. So then you have to analyze what is actually from these RNAs, what's coming into actual proteins, and then what are the reactions within organisms, and what's actually happening, and you can track what is happening through metabolites. So in case you would have all of this data, you can make much better diagnosis of what's happening, and find some interactions and something unusual. So this whole field is called multi-omics. If you combine all these omics and try to work with that and make diagnosis and find best interventions based on this omics data. And this is already not therapy. This is already more like diagnostics, right? So previously we were talking primarily about therapies. So this is first story about diagnostics. I mentioned this big ideas reports, which I sent to you from Katie Woods, from ARK Investment Fund, ARK Invest. This is one of the slides from this report. So it shows that, yes, we have 20,000 genes, around 20,000 genes. So when they are transcripted into RNA, we would expect 20,000 RNAs. But it turns out that they could be modified in some ways. It's called, so it's, yeah, they can be modified, can be spliced in different ways, etc. So there are around 100,000 already RNAs, ways how, like RNA variants. And then with that, we would expect that the proteins would be also 20,000. There are 20,000 genes which code proteins. We would expect 20,000 RNAs which would code then the 20,000 proteins, but it doesn't happen like that. Unfortunately, the RNA gets so there are different ways how you can actually transcribe into RNA, and there are different ways how you can translate it, and there are many like changes, and then the proteins may kind of assemble in a different ways, and then in the end you have 1 million of potential proteins within your body, and this is already complicated, but it's still feasible to compute and to find some kind of interactions and some understanding. So, and this is what omics kind of approach is doing, finding this relationships and finding how this works. So, so Katie Woods in her material, in her report, like makes a forecast that the space would grow substantially. And now it's around like around 100 billion opportunity, multiomics, and it will grow to 300 billion. So at least there's somebody who put the figure on paper. So now we can actually track that. I wouldn't, base any opinion on that. So, but if you are a startup, doing a startup, of course you like these figures. It's great. So you can show always there's this great kind of analyst and great actually money manager who put this figure, 300 billion, and I'm part of this story. So it's helpful. But of course, if in reality, I think that we may see a very different number. Could be much lower, could be not. Who knows? And finally, yeah. So these are like 3 dimensions of multiomics. I mentioned just one, which is this. So like now, hopefully you remember all of these words, no? Genomics, epigenomics, right? What else? Do you remember? Do you understand these words? No. So let's go. I'll go like quickly through that as well. Like just again. So everybody knows already this all these words. Okay. So again, so genomics is our DNA, is our genome, is all of our genes. Exons are those genes who are like coding actual proteins. Okay. So this is the genomics. Epigenomics is the coding which shows which gene to read and which gene not to read within a specific cell. Because all of our cells, as you know, hopefully as you understand, have the same DNA. But you have cells of skin, of whatever, of your nails, of like hair, anything. And so these are cells with the same DNA. But within this DNA, you have special marks which shows that in this particular cell, you read this gene and or not. And this is called epigenomics. So epigenetics or epigenomics, it shows all of these marks, and this, in the end, it's called epigenomics. Transcriptomics means all the RNAs which are being produced from, but actual RNAs, not which could be produced, but specific RNAs which are being produced in your body. This is called transcriptomics. Proteomics means all the proteins which are being produced in your body. Metabolomics, all the metabolites from all the chemistry reactions in the organism, and phenomics, phenomics is all the types of cells which you have in your body. So like phenotype, so you have genotype. All cells have this almost the same genotype. This all the same genes, but phenotype is what the genes are like off, on and off, and then by that they produce a specific cell which functions as it should function, like whether it's a skin cell or a lung cell or anything else. So this is first dimension. The other dimension is like organelle or like organ or organism. So this is the way kind of system biology works. And finally, you have to look into the age and environment. So, and all of that creates a multi-omics cube, and that's kind of an ideal kind of application, mobile application or anything, or kind of smartwatch application, whatever, where you can track all of these things, proteomics, genomics, and everything, and find some insights. The air is polluted like 20%. You should change, the country where you live. Or something like that, but because it affects you, and in a genome you have a predisposition for that, and in this country you may get cancer very quickly, etc. So something like that. You can get this. Ideally, at some point you will have that. Not now, but we will see who will do the startup. This would be a multi-billion opportunity, probably larger than Facebook or Meta. Okay, and finally, just another trend in multi-omics. Which is again, which like the same as Interspecious Communication, which I mentioned, kind of crazy company, crazy startup, which is kind of doing next generation sequencing, next generation microbiome. This is next generation of multiomics. So what this company does, it says that when you look into the cell and you look into RNAs being produced and DNAs and everything, it's very important where it's situated within the cell. So it's called spatial omics, spatial multiomics. So because if RNA, it's not enough to know that this RNA is being produced. If this RNA is not in the right place within the cell, then it's probably not functional. So it's called spatial multiomics. So you have to also look into where within the cell it actually is situated, so located. So this is the story, spatial multiomics. To finalize that, but don't remember that. Please remember this. Oh, sorry. So please remember, where is it? Yeah, this. This is more important. Yeah, this is a great chart here. Okay, so, and finally, our final topic for today. Because we have this spatial, whatever, multiomics, multi-omics, meta-omics, whatever, there's more and more data we get from that. So, and at some point, the data is to the extent that we cannot save it any longer. We cannot store this any longer. Also, if we talk about health, we want to store this data ideally, well, not forever, but until our death at least, right? So we want to store this data potentially after our death as well, so that we at least know what happened to us. So it means that there will be a lot of data which is being always saved, all these genomics, everything like DNA, RNA, metabolomics, proteomics, and everything. And then all the photographs of our lungs or anything like that. So like any of our organs, internal organs, etc. So at some point, this data is just too many, and we see exabytes and exponential growth of data. Of course, there's other things why it's growing because of video files, because we were previously were spoiled by Full HD. Now with 4K is not enough. Now we need 8K. All of that. So films also have to be stored for some time, right? So there will be a lot of data which has to be stored for a prolonged period of time, but potentially forever, but for tens of years. And the amount of data would be rising exponentially. You see here all of the devices, all of the whatever tests, everything which currently measuring us in where we want to save this data for future use. So because of this explosion, there are two problems here. Firstly, the currently available technologies, unfortunately, will never meet the demand for data storage. So I mean the currently available, which means the tapes, the hard drives, be all these physical ways how you store the data. Already 30% of data could not be stored in 2025. So the demand is rising much faster than what we can actually support. The second problem is that this is very energy energy consuming. So it requires a lot of energy. You know that these data storage houses are currently there, huge buildings with data storage, and they require a lot of power. It's already countries, the amount of consumption of power. So what can actually happen is the DNA. The DNA can store data, can be much in a very much smaller size. So it can actually miniaturize the hard drives which we have in today. The other thing is that DNA doesn't need energy. So DNA, they could be stored for centuries or potentially for thousands of years, right? And without any energy supply. This is amazing. So what we expect to see is that that DNA storage seems at least to be, for now, inevitable. It's just inevitable. You see here, how much already shown to how much we can store, right? So in a hard drive, and so this is the density of information and how much we can do. So this is currently available technologies, and it doesn't feel that they can be dense anymore. So it feels that there's a limit from the physical perspective, from the physics of that. And then there are new physical ways how you can store the data, but it's very exploratory, and it requires still a lot of work. But what DNA can do, it can be by multitude more dense on the one side, and it already showed that it could be scaled. So it feels like DNA storage could be the next big thing in the storage space. And of course, for DNA storage, you have to synthesize DNA. And with which technology you can do that? With yeah, with all the synthetic biology tools, generally. So CRISPR is more about editing, anyway. But, by the way, yeah, so I will show it to you. So here, yeah, here just for you, just some overview of major milestones in the DNA storage space. What I actually like about here is that it follows my my beloved logic that initially a technology would be tested in the art space, and then after it's been tested in the art space, it's been tested in the scientific, in the fundamental science space, and after that comes an entrepreneur who would combine that and make potentially a product out of it. So this was initially tested. Of course, they tried to encode the text, and they encoded Genesis. And they encoded Genesis and put this DNA into an apple. And apple would grow, and in each of its cell, in its DNA, it would have the Genesis code, the code of the Genesis from the Bible. So like essentially, the Genesis is about the apple tree at the very beginning. So this whole idea of putting this text into an apple tree, and growing this and making this an apple tree where inside is the Genesis code is very exciting. But then, of course, it gets more into kind of they actually coded Shakespeare there, and like some images, whatever. So this kind of so that's a great interconnection between art and science. But later on, they like saw that it's possible actually to code very like large files into DNA, and it feels like potentially DNA could actually be scaled. And these are already a few companies which are actually working on that. They are working from the biological from the angle, also working from the more engineering angle, like without biology, without kind of biology pores, more kind of engineering. So there are specific companies. They are backed already by by some large corporations like Microsoft and others. And this may be a big story. And there are some complications. So here, to finalize, so today, hopefully, you understood a bit more, some new things. Hopefully, heard something new. So today, we continue discussing the code of life, but now not about reading or editing. We talked about writing code. It is synthetic biology. And with synthetic biology, you can do really many things. And you actually can create life. You can synthesize life, but then you synthesize the software, the code of life, put this into like a cell, and then the cell would be acting as per the code. So this kind of a hardware. Hardware is the cell. And hopefully at some point we'll be able to actually construct the cell out of all these elements and have more control over it. We talked about microbiome. And this, hopefully you understand now how it works. You can restore the healthy microbiome, hopefully. Multiomics would also actually cover microbiome, DNA, all of these things. They actually measure all of these things and combine them and try to diagnose something which is happening, which is kind of wrong. So this is about multi-omics. And finally, we talked about DNA storage, which uses synthetic biology to store potentially multi-omics data. Thank you so much. Yeah, and now it's time for questions, if you have any questions. No questions? Questions. Okay, I'm always glad to hear questions.
Just a question of clarification. Can we go back to the previous slides with the chart of invests in synthetic biology?
Investments?
Yeah. Investments, yeah.
Did I have such a slide? I only have a microbiome.
No, there was something.
Yes, we have this one. Yeah, this
one. And here DNA data storage is zero, like zero, $0. What is it? Yeah, and the next slide is about that IT companies invest a lot in this area. I just missed the connection
Yeah,
between
yeah.
these slides.
Yeah, so DNA data storage is generally, yes, it's kind of a small still. And so this is potentially, I would say, this is our investment which we made. So this kind of, it feels like a Rna company. So the company which we invested into, DNA data storage company, the amounts look similar to like, it was 24 million round. But the Microsoft and others, they investing into different companies, not only into DNA storage by far. So the Bill Gates Foundation, I think, as far as I recall, I may be wrong, but I think they backed Twist. They definitely backed Ginkgo Bioworks, definitely. But Ginkgo Bioworks is not doing DNA storage, right? So Twist, I think, yeah, Twist. There were some big player in there, but probably not in the last year. So it's just just numbers for last year. But they are investing into I would say more kind of you see here the synthetic biology requires a lot of software capability and bioinformatics capability, and there are many companies which would be like not doing anything in the like wet lab, so not doing anything in the lab. They would be doing everything in silico, and this is where like Google and others feel comfortable investing into, or IBM, like IBM Watson, just for example. But anyway, so like IBM and others, they would be investing into this kind of stories because these are like they may apply their expertise in this kind of topics, and potentially through that angle enter the healthcare space, which may be larger at some point, than a pure IT space like social networks, etc. Yeah.
Yeah, okay, thank you.
But it's a very good question. I, I wouldn't spot that. That's a great question. Okay. Okay. Grisha? No? Okay, so next time we'll get more into digital space. So we'll get into AI drug discovery. So how artificial intelligence is being used for drug discovery. This is, again, one of the hottest area in the space. Actually, in our list of the hottest area, this was number one in terms of the number of investments and the potential deal values, etc. We'll also talk about digital therapeutics. Digital therapeutics is not telemedicine, and I'll explain you next time what it is, because people are kind of, for them, it's whatever, digital therapeutics, digital healthcare, telemedicine, it's all the same, but these are very different modalities, and we'll be talking about telemedicine, etc. here later on, but here we'll be talking about AI drug discovery, computational biology, and digital therapeutics, and then later on get into robotic surgery, cyborgs, medical devices, etc. Probably next lecture or the following lecture. Okay, thank you so much. Have a great day, and we'll meet on Monday. Thank you.